so I guess pass those on up if you're done. Come back. The extras. All right. So, sense of taste. Bet you got that one right. Gustation. Cell type makes new. Which cell type makes new olfactory and gustation receptor cells? Basal cells. So anytime you see basal cells, it's like a stem cell basically, right? You have the basal cells on the skin. You have basal cells here too. Not a cell type contained in taste bud. Is it olfactory gland cells, supporting cells, basal cells, or gustatory receptor cells? Right, olfactory smell, so you probably got that one right. Located in the back of the tongue. Is it valate, fungiform, or foliate? It's the first one, so the valate is in the back. And which is line provides a hole for taste dents to move through? It's the taste pore. See enough? Right. Sweet. There. Everybody see the final exam study guide? If you didn't, you know where to look for it. And we do have, obviously, Chapter 17, which is haven't been tested on already. So it is the whole study guide for 17. That's part of the final exam study guide. There'll be extra questions from there. Probably tomorrow I'll let you know exactly how many points for, all the old chapters versus 17. I haven't really thought about it yet. But there'll be, more because you haven't been tested on it yet. And then we'll do part 2 on Thursday, and then next Tuesday we'll have time to review that whole final exam study guide. So make sure you've got it all completed, circled the ones you looked back on, and you were like, I never really knew that. So if anyone looked at it, have you noticed that it seemed like it's a lot of the more complicated ones that are on there? So the ones I really want you to actually know when you leave. So might have lots to discuss, like how neurons transmit information and everything. I'm sure we'll be talking about that one. Right. So we've got, all these different senses, right? Vision, hearing, etc. And today we're going to do mostly sense of smell, a little bit of taste, and on Thursday mostly vision with a little bit of hearing, and because hearing's really complicated, so we don't really need to go into hearing too much. It's confusing. All right, so we'll do hearing or smell first. So it is actual chemicals. You may not think about it, but when you smell something, it is actually like a chemical going into your nose and doing something there. So kind of dissolved gas. And we have about, as far as we can tell, over 100 different receptors. And they're activated by different odors. And of course, an odor, what's different about different odors? They do smell different.
The chemical makeup of them?
Yeah, so the chemical makeup of them is different. So like, sulfur activates something, chicken, I don't know. Not sure which exact chemicals those would be, but they are just kind of floating in the air. All right, here's my little stick. So there's your nose, and the actual area where you sense those odors is way back in the back, the olfactory epithelium. It's kind of at the top of your nose there. And I guess an interesting thing when you're eating something, you do get odors coming back up from the back of your mouth too. As we'll talk about when we do taste. How does food taste when you got a really bad sinus infection? Right? Like you could just eat anything, it doesn't matter hardly. That's because most of your sense of taste is actually smell. Aside from like sweet, salty, umami, but most of like the finer things, which makes food actually taste good, is actually your sense of smell. So when people ask you like which sense do you want to lose, be careful. Because if you say smell, then you're not going to be able to taste your food anymore very well. So I'd go with taste if I was choosing one. But yeah, what would you want to lose? You have to choose. Taste, hearing, vision, touch. I think we could do without taste. That's funny. 3 types of cells, and then I list 4. Okay. So the receptor, the supporting, basal. These gland cells make some mucus, but not a whole lot of it. But it doesn't make most of the mucus. It's not like it makes all the mucus that you have coming out of your nose or something. It's kind of different. It's just a little mucus to dissolve things. But the receptor cells receive the signals. Supporting cells just kind of hold everything in place, support. And then the basal cells replace those dead receptor cells. Because you'll see this with taste too. All these receptor cells, they just kind of get, they get exposed to everything. So they don't live very long. So they're constantly being replaced.
So why do if you're used to a smell, then you don't smell it anymore? Like your car.
Yeah, so you mean like you get in, you go somewhere, you smell something, and then 5 minutes later you don't smell it anymore? Yeah. So there's probably 2 explanations. Well, 2 explanations. One is that receptor cell runs out of neurotransmitter to send. Or it could be that the brain kind of says, all right, we smelled it, like, got it, let's not worry about it anymore. And so it's not sending it to the conscious part of your brain. So it could be either one. Right? It's true, though. You do get used to it. Like and somebody will walk in the room and be like, oh, my God, what's in here? And you're like, what do you mean? All right, so if we zoom in, just to remind you, we're going to zoom in on the epithelium up here, so where we actually are absorbing those chemical odors and detecting them. And down here will be the part that's like exposed to the nasal cavity, so where those odorants are. You've got a little bit of mucus there. It's going to dissolve these chemicals. And then in blue is our receptor cell. And if you notice, they've got little cilium sticking out. So that's what actually binds to the odors. So like the chemicals have different odors have different shapes. These cilia have different shapes. Just like when we did enzymes at some point. Did we do that in here? Okay, so like maybe you have, let's pick one. Let's say sulfur's a square. What's something else you might smell? You think of? It could be stinky if you want, I don't care. Okay, so maybe it's something like ammonia gas. Maybe the triangle. So your receptor for sulfur smell is going to fit this one. So this would be the receptor. And then the ammonia one is going to be a different shaped receptor. So they just fit into that. Right. And the cilia is on the receptor cell. Right. They're little. You have microvilli in your detective tract, which are even smaller than cilia. Right. Yeah, except kind of the same idea. They stick out and do stuff. Except in the intestine, they're like catching nutrients and bringing them in. Whereas here, you're just kind of binding to it. And then we've got all our supporting cells, kind of your support. There's the gland cells that makes mucus. And if you notice, our basal cell sitting there waiting to replace this blue cell if it dies. What type of cell do you think the receptor cell is? So like an epithelial cell, a connective cell, a neuron, muscle cell. It's a neuron. And you could tell because here's this little blue thing. It's an axon. What kind of cell is the receptor cell? It's a neuron. So it's got its receptor, cell body, and then how do neurons send signals again? Between two neurons, synapses. What about how does it go from one part of a neuron to another? Like an action potential. So you start an action potential, it's going to travel. So there's your axons, and then it goes up to the olfactory bulb neuron, transmits the information, and then it's going to go through another series of axons up to the brain. Does anybody remember what part of the brain did sense of smell? What would be like the olfactory part, but which kind of broader part? So like cerebrum, cerebellum, hypothalamus. So it is the cerebrum, the sensory areas of the cerebrum. So that's eventually where all those signals are going. Because not until that signal gets to that area of your brain do you actually sense anything consciously. So, series of neurons.
So for those, you said that neurons don't usually, or they don't replace themselves.
We've been simplifying it a little bit. It's neurons in the central nervous system that have a hard time regenerating. So the actual neurons in the brain and the spinal cord, but neurons elsewhere can regenerate without too much trouble. All right, so which cell type actually makes our olfactory receptors? Let me start that over. Okay, people. I've got some Play-Doh today. All right, so it is basal cells, right? Let's be. So I guess I kind of already said this, and I don't think it's on your study guide either. But if you want to get like the broader overview, that's where it's being sensed. And then you go to the olfactory bulb, which is like right next to the epithelium, like where you actually sense it. And then right there is where those next neurons start, and then they travel up to the olfactory area in the cerebrum. Kind of see where those signals go. All right, so I think y'all needed some more practice on processes and one thing happened after the other. So I got a little activity for you to work on. And it's going to be basically how those receptor cells get to transmitting an action potential. Quite a few steps involved. So first, the odorant binds the receptor. Then that receptor, which is on the cell membrane, activates another membrane protein called adenylate cyclase. Adenylate cyclase makes cyclic AMP. Cyclic AMP binds to a ligand. I'm going to leave this up too. Binds to a ligand-gated sodium channel. What happens when you open a ligand-gated sodium channel? Sodium comes in. What kind of charge does sodium have? Positive. So what does that sound like to you that you've been learning about? It's an action potential. So you'd start that action potential, and then it would travel all through. And I'm going to put this slide back up, but just to give you an idea, you'd be starting your action potential right down there, right by that receptor. It's going to travel through that neuron to the next neuron, and then all the way to the brain. So that action potential you started travels through the first neuron and then through the second. Just like how you learned about how neurons transmit info. So one of the fun ways to make sure you actually know what all those words mean, because honestly that's a lot up there, isn't it? It's a lot of steps. So you're going to model it. And I've got the assignment somewhere in front of me. Yeah. So actually, let's just do number one first, go over it, and then we'll actually do the modeling part. So let's just do one first, about what all these things are. So what are each of those things? All right. Were there any that you had disagreements on? You're all good? All right, good. Let's do the hard stuff. So with your group, you've all got, and I was going to put Pedro and Cindy up here, make a group of 4. So I could do this together. So if you look at number 2, the next page, says you're going to use Play-Doh. You could cross out popcorn kernels. Using Play-Doh and cotton balls, you're going to create a model of an activated olfactory receptor and the electrical changes involved. So basically what you're going to do is turn these words into a moving model using those materials. It might take a while. So the first thing you need to do, though, is draw yourself a big cell membrane on your table so you can start adding stuff to it. I'll turn the light on. What? Oh, no, those are for you.
I know we all have different ones. I didn't see you give it to anybody else.
Oh, yeah. And it's fresh Play-Doh that I bought today.
Yeah. I'm not sure.
Well, so you want to draw it on your table so you can move Play-Doh around and cotton balls, etc. Did you get one? Oh, do them?
We're supposed to draw that, right?
Yeah.
Draw right here, that.
Oh, okay. I can't see that. It was
Oh, you're fine. What? Draw it on the table. Don't look at that picture. Because it gets you confused. Because they show it as like, because it's not moving. So that picture actually really confuses people. All right. So I really definitely encourage you for studying for the final too. If you're stuck on some process, just get out some pasta and Play-Doh and whatever you find in your house, and you could model out any of these processes. Yeah, your script would be this. Yeah, you don't have to turn it in. You could use beans, you could use all kinds of stuff. And if you notice what's cool about modeling it, did you feel like you were asking yourself a lot of questions or asking your partners like, what does that mean? What does that mean? It like makes you actually think about it or understand it. So, well, this one was better. Huh, interesting. Last time you said it, a minute ago you said it was Jordan's fault.
It was Jordan's fault.
Now it's the Play-Doh's fault.
I'd blame her too. Huh? I would blame her too. I would.
All right, so based on what you just learned, without looking at any notes, see if you can answer this. What's the order? What is the answer? That's happened before. All right, let's see what y'all got. All right, so maybe chat with your group. We're tied between C and D. Yeah.
Instead of like an extra test on.
Right, and tomorrow, the next day, I'll tell you exactly the breakdown.
Might not be organized by like chapter.
Yes.
Again, then I guess like
Yeah, I might put 17 first and then go 1 through whatever. Yeah. Yeah. All right, we got more people at C. So C is the right answer. What's a nerve impulse? Since that's what was different in C and D, it's what was confusing people. What's a nerve impulse? Yeah, so it's it'd be like an action potential going down the axon. It's like going towards the brain or something. And of course, you could put stuff in front of here, right? Like what happened before you made camp. But I figured there was enough words on there already. Couldn't really fit too much more. So if you're putting the whole thing, you'd say an odorant binds to the receptor, which activates vanillate cyclase, which makes AMP, and then you'd be there. So, all right, so why do you think different receptor cells sense different odorants? Oh my God. All right, let me fix it. Whenever,
If you just take out like the date.
there we go. That's what I meant, right? Okay. That's what happens when you like copy and paste and don't read over it carefully. What do you think? All right, so tie between E and F. Almost exactly. So convince your neighbors that you're right. All right, final votes. All right, still almost tied. So the answer is E, A and B. So basically everybody agrees that A and B are right. It's just a matter of whether C and D are too. Different receptor cells let in different kinds of ions. No, what ion do they let in? It's all sodium. So the only thing that's different is the actual receptor, the olfactory receptor. So that very first step where you drew that receptor, but it all leads to making CAMP, which opens a ligand-gated sodium channel. That happens in all of them. So for example, some people had like a triangle-shaped receptor. Somebody had like a couple, like a 2 eyeball-looking thing back there, receptor. And I know D tripped you up. Were you thinking this was talking about the receptor? Because it says the cell. So the cells are the same size. Maybe the receptor is a different size. But all those little blue cells in that picture, they're all the same size. That was a little bit mean. All right.
Is it possible that if you sense so many different odors that there's a few different receptors?
That's funny because that's question 3 for your group. Thanks for the segue. Did you actually read that off of there, or did you think of that? Yeah.
Well, it's a study guide question.
Yeah.
So
Yeah. So have you chat with your group about that, see if you can brainstorm some possible explanation, and then we'll go over it. All right, so that was just a brainstorming. What did some people come up with? I actually heard some correct answers out there. Who wants to share their idea? Okay, so yeah, that's the basic answers. Different combinations of the different receptors. Because you've got maybe a couple hundred, but think how many combinations that gives you. Because it could, it could be 12 of those 100, or a totally different 12. And so you get these raw signals, they go to the brain, and then that association area says, well, when these 12 are activated, that's pizza, or that's cookies, or that's a bathroom, something like that. You don't have pizza receptors, bathroom receptors, and cookie receptors in your nose, at least as far as I know. And actually, I just realized I don't know what actual smells you have receptors for. I'll have to look that up.
So what if you install like a new smell? Would just make a different set of receptors, like a different combination of them?
So if it was something you'd never smelled before, you would store it as like a memory in the association area after you figure out what it was. So you could imagine like when a baby's smelling things for the first time, they don't know that that's pizza. So you like tell them, or they see it again and they know that that smell goes with that food or something. Kind of interesting. And they actually think that babies smell their mom. Like that's how they know it's you. Because their eyes, like, I think y'all probably know that. Like they can't see very well when they're really newborn. Definitely. So it makes sense that we would do it too. Kind of interesting. All right, so basically just different combinations. So it's kind of like the same thing of how you get different colors, right? There's only 3 primary colors, and put them together all different ways, and you get like limitless colors, right? Same idea with the smell. All right, so what about taste? We won't do as much on this. There are 5 tastes: sour, sweet, bitter, salt, and the new one is umami. They kind of finally proved that exists. That's what MSG activates, or basically anything meaty. So like why steak tastes so good, or like a big bunch of salmon, like fatty salmon tastes so amazing. That's the umami taste bud. But if you notice, these aren't all that specific, right? Sweet, bitter, it's not all that specific. So the way your food tastes is a combination of those plus all the olfaction, all the smell. Because when you're chewing it up in your mouth, it is going back up through the sinuses. Right, so combinations of those, plus I would include the olfaction. Because your brain can't actually, the conscious part of your brain doesn't distinguish. Like you can't tell, oh, that part of the taste is from my taste bud and the other part's from my nose. Right? You can't tell, can you, when you eat something? You just know it tastes like this. You mean when you get older? I think you get less of certain ones. Or at least that's what I've heard from people. I'm not sure exactly how you test for that. Or you could just get used to it. Maybe you just decide that it's a good taste. All right. Just want to show you a few things. Your taste buds aren't actually directly on the surface of your tongue because there's too much flow of things and saliva moving around, they would get damaged. So instead, there's this little taste pore with these microvilli that bring the cells in a little bit. So you can see how like the taste bud itself is inside a little bit. So whatever you're eating kind of goes into that channel where the actual taste bud is. They're replaced every 7 to 10 days, your taste buds. So these little actual, what are they? The actual receptor cell in blue there, they're replaced every 10 days. Which is why if you ever burned your mouth in like tea or pizza, so I'm talking a lot about pizza today. But that's like, I don't know, that's the worst way I've burned my mouth. It's like you bite into pizza and hot sauce goes everywhere. Did you notice like your sense of taste was kind of messed up for a while? That's because you burned those cells and they died. And then you just replaced them.
How does it work if it's like not something hot, like if it's something really spicy?
You mean how do you taste it, or does it damage?
Sometimes it like really burns, you know, if you eat something really hot.
Yeah, so those are like actually probably damaging those cells. And you do have pain receptors in your mouth, which are what spice activates. It's actually pain receptors. So if you notice, there wasn't anything for spice or spicy up there. So it's actually like a pain receptor.
So if you have, so if you like really picky eaters, is that more psychological?
I mean, it's hard to say exactly. I would think so. But how would you prove that someone doesn't have different taste buds? It's sort of hard to prove that, right? Because you always hear that from people like, oh, I just have a strong bitter taste bud, or, you know, a sweet tooth or something. But most likely it's your brain, I would say. All right, so there's the different areas we talked about. You did on your quiz. And it's kind of interesting. So your taste buds, they don't work quite like the olfaction in terms of how they start an action potential. So the salty ones, what's salt? Sodium. So it's actually, those taste buds just have sodium channels in the membrane. So sodium actually comes into the cell from the salt. There's none of this receptor, denylate cyclase, none of that. It's just you've got a channel for sodium, it comes in. What does that do to does that make an action potential? It's very direct. Same thing with sour. What do things that are sour? Are they acidic or basic usually? Acidic. What do acids have a lot of? What molecule? Hydrogen. So look, we've got a channel for hydrogen ions in our sour taste buds. So if there's lots of lemon juice in there, there's going to be lots of H pluses in your mouth that are going to go right into those sour taste bud cells. Which, does that make an action potential? Right, it's positive. So that would make one too. Sweet, bitter, and umami, that's more like the olfactory ones, where there's like a receptor for those certain things. Activate a few molecules, open up a channel. All right, almost done. Let's see, I wanted to go over this one. So, have you seen this map before of like where your taste buds are? No? No? Okay. All right, do you think it's true? A for yes, B for no. Sorry, this is a little bit different of a question. Let's head out. Great. So we got 58 for no. And it is not true at all. So it was something that they thought they discovered in 1906, and then no one really studied it or tried to prove it wrong till like 1975, which is crazy because how hard do you think it is to test if you can taste salt back here on your tongue? Like, how easy is that? You just stick your tongue out and put it there. And actually, I think I learned this in like middle school or high school. I learned this. And actually, they still teach this. But it's so easy. Like, well, I can taste sour at the tip of my tongue, can't you? It's very strange to me. But still taught a lot of places. Test it out. The only one it looks like, yeah. So anyways, kind of silly. All right. So that's it. We're good. So we'll do part 2 on Thursday.